Since chimpanzees and humans are predicted to have only diverged 6 million years ago, there shouldn't be a difference of thousands of genes between them. 6 million years is just not enough time for a significant change like that to happen. So what does this mean? Does the missing chromosome in humans disprove the theory of common descent? Well, no. As it turns out, this missing chromosome is actually a powerful piece of evidence in support of the theory. While completely deleting a chromosome has huge consequences, there's another way that the 24th chromosome, present in chimps and other apes but not in humans, may have gotten lost. Under certain circumstances, two chromosomes can become fused without changing how their genes are processed. The location of a gene on a chromosome doesn't change how it's expressed, so sticking two chromosomes together is something that could easily happen within 6 million years. So the mystery of the missing chromosome gives us a really elegant way to test the theory of common descent. If Darwin's theory is correct, we would expect...
But okay, so maybe I'm just a layman on this, but this seems to me if there was a fusion, wouldn't they both have the same amount of chromosomes?
No, so what they're saying is and Jeremy, you can explain it too. So the chimp human common ancestor, let's say 6 million years ago, would have had 48 chromosomes. Today we see humans have 46, chimps and the other great apes have 48, for example. So, why this discrepancy in chromosome number? So they, predicted that maybe there was a fusion, for example. So the human, the chimp line split, boom. Chimp line goes one way, human line goes another way. At first, whether it was for 1 or 2 million years, you can never get a straight answer from the evolutionists, but both lines would have had 48 chromosomes. But at some point in the human line, there would have been this chromosome 2 fusion, and then it would have had to been passed on, they'll say, through genetic drift to every single human on the planet. Therefore, we now have 46, and the chimps and other great apes have 48. So no, we wouldn't expect the same number because the fusion happened after the split in the human line. Does that make sense, Praise?
Yeah, I think so.
Yeah, but wouldn't the information be like? Wouldn't that make it like a bigger chromosome?
Like the fusion? Well, they'll say that the telomeres at the ends of the chromosomes would have degenerated because the area is very degenerate. So that's kind of their explanation. But here's the thing I want to point out is that question you made, Praise, actually brings up another point. Is they're looking to the so-called chromosome 2 fusion as one of their better lines of evidence for human ape common ancestry, but even they will admit that this occurred in the human line. Therefore, it's indirect evidence because it's not like it happened before the split, which means even if there was a human chromosome 2 fusion, it could have occurred pre-flood or post-flood around the time of Babel, before the worldwide dispersion for everybody to get it in time. But the evolutionists would have to explain how they could have, a bottleneck, or how genetic drift could make every single human being on the planet have the chromosome 2 fusion. So, I mean, me and Jeremy are about to get into the discrepancies showing that it probably isn't even a fusion. But it's just funny because even if it is, even if you gave them the benefit of the doubt or agreed to disagree, it's easier for us to explain a fusion in the human line because we have a couple bottlenecks in our model, the flood and Babel, where it could easily be passed on. They have trouble explaining how it could be passed on. I've asked Conspiracy Cats that, and he kind of avoided the question because it's not an easy explanation for the evolutionists. Go ahead, Jeremy. I know you're going to say something, brother.
Yeah, I'll just make it short and in a nutshell. As we know, telomeres prevents fusion. Okay, that is one of the evolutionist problems, and they got rescuing device for that. And those telomeres are, like you said, did you say it? These telomeres are so degenerate in the fusion site itself. Okay, and why are there and there's lack of telomere in those sites also, and why are they so degenerate? And don't forget that if you say degenerate, it doesn't mean sorry. If they are degenerate, it means there are not enough telomere sequence as well.
Right.
Okay.
Well,
This helps.
Well,
Yes.
and I'll just play devil's advocate. So, because I've debated, I mean, Erica, I think we had 32 minutes worth of chromosome 2 fusion talk in our last debate. So they'll argue that, okay, since healthy telomeres help to prevent end-to-end fusions, okay, these telomere to telomere fusions, like you were saying, they'll say that both of the 2 fusing chromosome tips, these telomeres, for example, were severely degraded, with possibly even both of the telomeres degraded to the point of near extinction. So therefore, they're not able to do their job now sufficiently. So bam, you get the telomere to telomere fusion. So how would you respond to that, Jeremy?
Say again. You mean the ends, right?
Well, because the job of telomeres is to prevent fusion. A healthy telomere, that's its job, right? It's the end caps of like think about the ends of your shoelace, for example, right? Well, over time, what happens, that little plastic piece kind of degrades and it starts flaring out, for example. That's kind of like the telomeres, right? Ultimately, the telomeres shorten over time. So you pointed out that, the job of telomeres is to prevent fusion. And I always ask that too. My first debate with Erica, I said, okay, why were the telomeres involved in a fusion if their job is to prevent fusion? That's usually a lead-in question to see if they know what their next argument is. Because if they don't, well then boom, the debate stops there. But typically, so a more informed evolutionist will respond with, okay, yeah, you're right, that is their job. So they're proposing that those end caps, those telomeres, were degraded to the point of almost near extinction, which means now they can't do their job because they're not healthy anymore. Therefore, they were able to fuse. And the area is so degenerate now because the telomeres before they fuse were already degenerate. So how would you respond, Jeremy?
A degenerate telomere cannot perform a fusion. Have you heard of this standing for truth?
Well, I've heard that the telomeres prevent the fusion, but if they're degenerate to the point where they can't do their job, now there can be a fusion that takes place.
One point to make would be that without this fusion, evolution would be false.
Right. So, so here's the thing. So that's their answer. So my question typically is, okay, I think that that's weak, but it is theoretically possible. But I would say, why would both of the fusing chromosomes be degraded to exactly the same point, literally yielding a minimized semi-symmetrical fusion site? So it's kind of a stretch, but the fact is the area is degenerate. Oh, go ahead.
I remember. Sorry. I'll let you finish.
Okay, so I was going to say, so yeah, that's what they'll say. They'll say, okay, so the area is degenerate because those telomeres at the time of the fusion would have had to been degenerate anyways for the fusion to even take place. Or else there wouldn't have been able to be a telomere to telomere fusion. But so there are some sequences there. They'll point to, I think it's like a 798, and I should pull it up, but I think it's about like a 798 base pair region. So there are some what looks to be like some internally telomeric like repeats. But what's funny is those internally located telomere like repeats that are at that region, they've actually been found to occur throughout the entire genome, from only just a few repeats in length to literally hundreds of repeats long, which and these repeats are not even associated with fusions in these areas. And there's a few papers that suggest that maybe they're involved in like expression, and maybe some of them have multiple functional roles. So yeah, those telomere repeat sequence. So even if you just say, okay, fine, I find that your explanation is weak, that those telomeres were degenerated to the point where, okay, boom, fusion. Okay, now let's look at that degenerate region. Well, those so-called sequences are are distributed widely over the human genome and not necessarily unique to fusion events, and there's good evidence that there's activity in those regions and possibly involved in expression, for example, like they're binding to things called transcription factors, which then initiate the copying process of a gene into RNA. So that even their rescue device is easily refuted, is my point. So I know I talked a lot there. The floor is yours, Jeremy.
In short, we found out that they are not as degenerate as their explanation is not as accurate as the data that we have found out. We found out that those end caps that you're talking about is not as degenerate as they are claiming. That's the answer, right?
Actually, say that again. Are you saying the telomeric regions are not as degenerate as they're claiming?
Yes.
No, they'll admit that it's degenerate because 798 bases that are seen there, it's an extremely tiny signature for an event, but their explanation is okay. They have to be. Is there some back, Jeremy? Is that background noise coming from you, brother?
No,
Looks like
Gavin.
So it is. Gavin, you got some background noise coming from your end, brother.
Look, I'll mute myself, and there's no noise from my side.
No, it's fine. It's fine. It was coming from, and it is good that we spend some considerable time on this fusion event. So we haven't even gotten to the fatal blow. So one major problem. There's multiple major problems that they have. So for one, those internally located clusters of what they say are the leftover telomeric sequences from the fusion, they're a lot more common than people realize. And they're found throughout the genome, evidence that they perform functional roles, for example, expression-related roles, and they're not necessarily indicative of a fusion, for example. So on to the next. The one fatal blow that I typically bring up in discussions, I brought it up to RJ Downer recently. He had no explanation for it, is the fact that there's these 8 specific satellite DNA sequences that are found in 8 chromosomes, for example, but this specific satellite DNA is not found flanking the human chromosome 2. There's a paper, it's the Ventura paper, that goes over this in great detail, because the presence of that chimpanzee and gorilla specific satellite DNA sequence in their genetics, but completely absent in humans, is not only a major problem for the fusion model, it's also a major problem for human evolution to begin with. Now, what's funny is the evolutionists will say this, okay, so why is this ape-specific satellite sequences, why are they absent in man? Okay, because realistically, there should be some of these sequences found flanking the reputed fusion site if we're related to apes, okay? So they'll say, well, it's been maybe 3 or 4 million years since the fusion, so maybe due to mutations or selective pressures, those satellite sequences that were once there at the time of the fusion in the human line, maybe they were lost. Or maybe the sequences were never there, but independently in the chimp lineage, they had some type of de novo origin. So what's funny is either way, they have a rescue device, what's called time did it.
Or there's one more possibility. Maybe we don't have the same common ancestor.
Exactly. And that's the one they don't want to admit. What's fun, because think about it, and you got somebody like ESE, emotionally stunted emoticon, he doesn't understand it. Like, why would we expect those to be there, right? Think about it. Even according to their model, okay, 6 million years ago, you got the chimp-human common ancestor, okay? This common ancestor would have had this very distinctive and very large sets of satellite sequences at the ends of their chromosomes, okay? So now the 2 lines split. Boom, you got the chimp line going one way, you got the human line going the other. both lines still have these very large satellite sequences at the ends of their chromosomes. Then all of a sudden, the fusion happens only in the human line. Okay, well, the satellite sequences should still theoretically be there. Now, at some point during that line, they would have been lost because we don't see them. But they won't even question the theory because guess what? Maybe humans and apes aren't even related, and all it is a rescue device. They have no predictions as to how much of this would have had to been lost in how much time, for example. And here's what I really find funny, and I pointed this out to James Downard. If this ape-specific satellite, these sequences were found flanking our chromosomes and more specifically the reputed fusion site, what would they say? They'd say, oh, here's more evidence for the chromosome 2 fusion. They'd love it. They'd brag about it. They'd talk about it in every debate. But since it's not found, now they have to say, well, why would we expect it to be there? It should have been lost. So do you see how no matter what the observation is, they're just going to force fit it into their stupid little philosophy?
Eventually, when they compare, when they're able to compare everything, they're going to be able to identify a specific human genome and that the chimp genome is not the same genome. Basically, sorry.
No. At any point, you guys.
Standing for truth, you have explained it very well. The satellite part, I like that. I heard that before from you. But since we are making a comprehensive video here, may I complete the entire argument, the simpler ones for the audience?
Of course, yeah. Chromosome 2 fusion is a major one they use, so this one deserves a lot of time, to be honest with you. So let's be as comprehensive as possible. So go ahead, man. Whatever thoughts you have, let's demolish it.
Things that came to mind. Okay, the gene which say prove the fusion, you know this, does span the fusion site. Okay, I mean this chromosome here, and then this the chromosome here which fuse. There is a same pattern of genes from this side and the other side. So if a gene is unique on the site before, and again I said before the fusion, then why do we see them on the other side of when the fusion happened? Okay, it means it's just one chromosome gene. It's just one chromosome. Okay, because you see similarities and uniqueness between those two fused chromosome. Okay, so it's just one chromosome when you incorporate this evidence, and they have rescuing device, of course. And there are more chromosome samples of this. It's not only chromosome 2. Okay, you can even go look at the straight lined version of the chromosome, compared it side by side. There are also not enough telomere, by the way, in the middle of the fusion site, as you know, like lack of glue to glue two things. Okay, like for the audience, it is like the fusion site is like a glue. Okay, and there's a lack of glue to glue the two things. That's the way I see it. Agree? Do you agree, standing for truth?
100%. Yeah.
Okay. To finish this off quickly, all right, and some don't even look like telomeres, right? Remember that? Okay, those telomeres. Some don't even look like telomeres. So if fusion really happened, and now we're talking about centromeres. Centromeres at the I wish I could share something here. I wish I prepared a slide or something. I didn't really prepare this anything. I was sleeping.
No problem. And that's what's good. We don't know what we're expecting. We just get it. Oh, go ahead, Jeremy.
Yeah, if a fusion really happened, now we're first we talk about the telomeres, standing for to talk about the more complex stuff, the one that really evolutionists can answer. But what I'm saying here is the things that evolutionists can answer, but they can answer, but it is just a tie. It is not really disputing what I am saying.
Right.
Okay, it is just based on worldview. So if a fusion really happened, the centromere now, sorry for repeating myself, I'm trying to speak English quickly.
Of
I'm
course.
weak at English. The centromere at the upper right side of the the fusion, okay, up the at the upper right, it's really hard without any slide to show. The centromere at the upper right side, at the upper right side, after the fusion line, centromere should still be active, but it became inactive, which goes against fusion because it's a line, right? It's like a pseudogene that it's just an example of the pseudogene. The pseudogene should have been passed, so this centromere should also be passed. Okay, and we do find the lack of centromere in uh, in the line where setting for two. Do you understand what I'm talking about?
I do. You're
Yeah,
doing good. Keep going, brother.
yeah. I really wish I could share a picture here. Okay, okay. So like I said, after the fusion line, centromere should still be active, but it became inactive. So it's like a pseudogene.
Well, the
Okay.
centromere, well, and just so the audience knows, because two active centromeres would be detrimental to the organism. So when the fusion took place, one of the centromeres would have had to be deactivated. Boom. Now we only have one active centromere. So now they need to identify in that reputed fusion site region evidence for a deactivated or cryptic centromere.
Yeah, and what we said is breaks the line, correct?
That's right. Well, they do
It's like,
point to an area. They do
no,
point to an area that they say is the cryptic centromere.
yes, so that is their worldview. So like I said, what I'm saying here is the more simple stuff, and they have their worldview, but what you said a while ago, they cannot answer. Okay,
Right.
so it's like a pseudogene, for example, that is supposed to go all the way, okay, but this centromere has kind of like what's the English for it? Has kind of what is the English standing for? Truth? What you said? The stop. It
Cryptic,
stopped.
a deactivate.
Yeah, it was deactivate. Okay,
Right.
so that's not supposed to happen. Okay, so after the fusion line of the centromere was compromised, so not compromised, but cut or deactivate. Sorry for my English. Cut or deactivated, where they should be active, but it became inactive, which goes against the fusion, right? You agree? It goes against the fusion, right? So, right?
Are you
Yeah,
wait? Are you saying that one of the centromeres that are deactivated goes against the fusion?
yeah, because it breaks the line.
No.
Okay, because there are
No.
several centromeres.
No.
It breaks
Because
the line. It
if two
after
chromosomes fuse, if two chromosomes fuse end to end, a telomere to telomere fusion, like they say, there'd be two activated centromeres present, but that would be detrimental to the organism. Therefore, one of
no.
the centromeres would have to be deactivated.
Yeah, there are two centromeres over there. If
But
you're
one of them would be
one is active and one is deactivated. Remember?
right. Yeah.
Okay.
But that fact wouldn't be inconsistent with the fusion. That's what they would expect.
Really?
Of course, yeah, because two active centromeres is detrimental to an organism. So they say that the second centromere, the cryptic centromere, I say cryptic because that's the one that it's like, where's Waldo? Where's that second centromere if there was a fusion? It would have to be disabled. So they've pointed to a region, okay? It's about 170 base pairs long. It's a repeat. And it characterizes primate centromeres. And Tompkins has pointed out that that region that they point to that is supposedly that second centromere, except in a disabled form, is way too small. It's too human to actually be a remnant centromere. And this entire sequence, okay, that they say is the so-called disabled second centromere, this is the fatal blow, okay? It's internal to a functional gene, just like the so-called fusion site is internal is a promoter for a functional gene. So that would actually be impossible if the so-called region there was ever a real centromere. So that just completely destroys their idea that it's the remnants of an ancient centromere. And so now what's amazing too, because now we still have those sequences we have to deal with, okay, that region, that 171 base pair repeat, just like the region with the telomeres was highly degenerate, okay, but there's still a region we have to deal with. Well, the telomere-like sequences is not actually the remnants of an ancient fusion. They're found all throughout the genome with strong evidence for function. Now, these alphoid sequences found at the so-called cryptic centromere site are actually a type of sequence commonly found in the human genome, just like the telomere-like sequences, outside centromeres. And they're labeled as higher order repeat. So yeah, this is the fatal blow. And for the audience, if you ever go up against somebody on the fusion, the fact that the cryptic centromere is actually a functional region inside a protein coding gene, okay, encompassing both protein coding and non-coding regions, is strong evidence that it is a key genetic feature to our genetics and not some leftover remnant or some cryptic disabled centromere. So every single one of the observations that should be found, okay, remnant telomeres, a second disabled centromere, they have all been overturned. They've all been refuted. The satellite DNA that's found in chimpanzees and gorillas, but not flanking the reputed fusion site, that absolutely destroys the model. And me and Jeremy haven't even gone over the fact that, well, we did kind of briefly, unless you have something to add to that, Jeremy, but did you want to go over the fact that the reputed fusion site itself is actually overlapped by a functional gene? Did you want to go over that fact?
At the fusion site itself? Is that what you are referring?
Well,
The degenerate site is overlapped by a functional element, right?
That's right.
Yeah. I haven't studied chromosome 2 for a long time. It was you who made the better argument that I made. But I have last thing to say about this chromosome 2, which came to mind right now. Just one last thing. There was a prediction before that we should find more centromeres in certain areas. Okay, but the prediction failed. It's lacking. Heard of this? Standing for truth? There was a prediction that there should be more centromeres in certain areas.
I've never heard of that. There should be more centromeres where in the gene in that area.
In the yeah, I wish I really have a picture here to show.
What is a centromere? It's the center of a chromosome, and the telomeres are the ends of the chromosome, right?
The centromeres are, yes, the center of the chromosomes, and at the center of the chromosome is the telomere, which they say was a fusion site.
Right. The telomeres are the ends of chromosomes. So therefore, when they fuse together, okay, so you picture one shoelace and then another shoelace with those little plastic ends, smash them together. So now you should expect to find those ends now in the middle and not on the ends. So that's what they expect to find, the telomeres in the middle of the site. So the region they point to, as we pointed out, is degenerate. they say, well, we expect it to be degenerate because it had to be degenerate for there to be a fusion. And we say, okay, fine. That's a rescue device. It's kind of a tie at this point. But the fact that that little tiny degenerate region is actually found all throughout the genome and found to be functional in expression, for example, that just shows that these are functional DNA elements and not remnants of ancient fusion. And the centromeres, there should now be two, one activated and one deactivated. So I'm not so. Why would they predict there to be more centromeres? I don't understand that one.
The centromere that you are talking about is from this is. Can you see my hand here? This is the first chromosome, right? And this is the second the chromosome. Okay. There's just a fusion site over here at the middle. So there's a centromere here, and there's the centromere here also. One is active and one is inactive. Okay. The prediction was there should be more centromeres. Okay. In that line. Okay. But it's lacking. Okay. You heard of this?
Are you saying centromere-like sequences, or actually more than two centromeres?
More than one centromere, because this is the last argument. Because this is the last thing I. Sorry about the sound. I think you guys heard it. Yeah. Where was I? I really want to make this quick so that the others can speak. Yeah, centromere here inactive and centromere here inactive. Okay, and in the middle is the telomere. Okay,
well, how many centromeres are in chromosomes? Because I always thought that the centromere is at or near. It doesn't always have to be right in the middle, but
in the middle, yes, in the middle of a chromosome, yes.
Right. So if two chromosomes, if two chromosomes fused, so now that chromosome 2 should have two centromeres, one deactivated and one activated. So you're saying that they thought there'd be more than just two?
Yeah. That was their prediction. You can check Dr. Tompkins' data. I guarantee standing for truth. The prediction was that we should find more. The prediction and again, there is a prediction that we should find more of it on certain areas, but it's lacking. Okay, I promise you. You recheck Dr. Tompkins' work. Even Dr. Even Dr. Georgia Purdom agrees. Okay, I think
I think what you're confusing, Jeremy, is okay. So you have a centromere. Okay, so now if there's a fusion, you'd have two centromeres. Therefore, if there should be an area of a deactivated centromere, but what is found is far too small to actually be like sequence-wise, because what they point to is 171 base pair repeat region, and Dr. Tompkins points out that area is far too small and far too human to actually be a remnant centromere. And the fact that entire sequence is internal to a functional gene really tears that apart. But so I think that's what you mean. I think the region that should be a deactivated centromere is too small, is what Tompkins points out, to actually be the remnant of a centromere. Is that what you mean?
Almost. Thank you for clarifying, my friend.
No problem, brother. No problem. That's what I'm here for. One last point on the fusion.
That will be all. That will be all. That will be all. I have nothing left to say. You have provided. Yeah. Okay. That will be all. Good job. Good job, my friend.
Thanks, brother. You too. You too. So I want to point one last part. So the fact that reputed fusion site itself is overlapped by a functional gene means that so-called reputed fusion site is not a fusion site at all. It's a functional DNA element or a functional component of what they call the DDX11L2 gene. And that so-called fusion site lies within, and you pointed this out before, the first intron of that gene. Have you heard about this argument, Jeremy? So there's been some criticism, though, and that says, okay, if that reputed fusion site really is just a functional region, okay, they'll try and downplay the significance of the of what Tompkins points out as promoter function and activity in that region. He will, well, a lot of critics, they'll compare the activity of the non-coding DDX11L2 gene to that of protein coding genes. And they'll have these diagrams up showing expression levels. Conspiracy Cats did it to me. And they'll say that the expression levels are, and the activity in the fusion region, which is non-coding, we know that, is extremely limited compared to the protein coding genes. So they're trying to say that it's not functional. Have you heard that before?
So you're talking about the, let's say this is a fusion. Okay, there's introns and exons on both sides, right? That's what you're talking about, correct?
Right.
All right. So yeah, I agree with everything you said.
So they'll say that it's not okay. So here's what Tompkins points out. He points out that these critics don't realize that long non-coding RNA genes, they cannot be properly compared to protein coding genes. And here's why. It's because long non-coding RNA genes are expressed at much lower levels. Therefore, it's not good method to compare those and try and say, hey, listen, you know, this area has low expression levels, therefore it's not it's not, there's no activity or function there. So that biological importance can't actually be measured simply by, which the critics do, comparing them to protein coding genes. And I like the way Dr. Tompkins points out, he says, and I'll just quote it, he says, this is an important detail that ill-informed critics don't realize, because he's compiled data that includes epigenetic marks, transcription factor binding marks, transcription start site data, expression levels, co-expression data with other genes. So Dr. Tompkins points out that all of the genomic data in combination, when you look at it as a whole, which is contradictory to what the critics are doing because they're so desperate. They're so desperate to hang on to this being an actual fusion event. When you look at it all in combination, they all corroborate on one clear fact that actually demonstrates that this alleged fusion site really is a second promoter in that DDX11L2 gene. So boom, we've destroyed the telomere-like sequences that should be there. We've destroyed the fact that there should be a cryptic centromere. We've destroyed the fact that there should be some ape-specific satellite DNA flanking the reputed fusion site. And we've shown conclusively just how functional this region is. So they're really hanging on to very little here, Jeremy.
Don't forget the synteny thing, which is on our side. You know about that, right? The
Which one, brother?
Thing.
The synteny.
The synteny thing.
Yeah, you know what?
Chromosome, the
Right.
synteny thing.
Hey, can you cover that one? Just so we can, like you said, make it as comprehensive as possible.
Yeah. It is inconsistent. I really. Do you remember the exact data about this standing for truth? I'm kind of lost here.
Oh yeah, no, don't worry, brother. You can leave it to me.
Yeah,
This issue.
you do it. I'm kind of lost regarding this, but I remember there's one more. It's the synteny thing, okay?
Right.
Because I think we want to make this comprehensive. So I recall there's the synteny thing also, and I think that's the last one.
Yeah, and it's the fact that there's no homology where there should be on the fusion site. There's lack of homology is what you're getting.
Yeah.
What
Yeah,
you're
thank you for
getting at.
reminding me.
No problem.
Thank you for reminding me.
No problem. I just think it's funny because they'll say this is in praise. Just think about this. They'll say this is a fusion site. Boom. Telomere to telomere fusion. Smack them together. Well, guess what? You don't achieve incredibly important genes and incredible function by just boom slamming together two chromosomes. So, but most importantly, even if there was a chromosome 2 fusion, they will admit that it occurred in the human line. Therefore, it's indirect evidence. And think about this. Let's just say there was a fusion, okay? Well, the fact that there's no ape-specific satellite sequences found flanking the region, okay, that's, and Dr. Sanford brilliantly pointed this out. He said, fine, fusions in general are evidence for genetic entropy because fusions, believe it or not, are actually mistakes. You can get fusions in cancer cells, mistakes in the crossing over process, for example. I mean, these are evidence and demonstration of the fall in genetic entropy. So even if there was a fusion event, since there's no ape-specific satellite sequence DNA there anyways, okay, it's evidence for a human-only fusion event unrelated to chimps and humans, and we can explain it better based on either post-Babel or post-flood, since we have two bottlenecks. So for the creationists out there, don't be scared of this piece of evidence because there's a lot of data, as Jeremy and I covered, that refute a fusion. But even if there was, don't be scared because it could easily be explained according to our model. Did you have anything to add there, Jeremy?
I think we covered everything, so we can move on. Good job.
Thanks, brother. Good job to you too. So I know we're being comprehensive, but I learned that from Jeremy. When it comes to their best evidence, Jeremy, what do you say? You got to be comprehensive, right?
Yes, just like the gravitational time dilation and the nested hierarchies we were talking about yesterday, the other day. And this video also talks about pseudogenes, as I recall. So nested hierarchies, in a different point of view, we will have to discuss when we reach that point later.
Right.
Yeah.
In the video.
They always talk about reutilization of the genome. They always talk about the repurposing of genomes. For example, they say that copy was made, or in this example of the fusion, they say that it was repurposed, but they don't really have any evidence of that ever happening, do they?
Sorry, repurpose in English is what?
Like they'll say you make a copy of the genome, you have an extra copy of the same information, and then they'll say, well, that information is going to be repurposed for some other use, but that's not really ever been observed. They're just making it up, really.
So you're talking about gene duplication here, right?
Yeah, and in this case too, where they put the two chromosomes together, and then now you have a functional gene in between the two chromosomes, and they would have to say that was a repurposing of the genetic information.
That's a great point. Yeah, that's, it's good. I'm glad you brought that up because that's where their philosophy comes into play, right? That's what they always look to the repurposing of certain sequences. Yeah, so they'll say, I've had them say, I think Erica too, like, so what if the region's functional? Is it impossible for that region to become functional at some point? Just like the ERVs, the ALUs, the retrotransposons, the pseudogenes, you point out the fact that these are incredibly functional to healthy life processes in the cell, for example, and in the embryo and stuff. And they'll say, well, as you said, jungle jargon, these functions or these genetic elements have been repurposed to be functional, or these functions have been co-opted. But once again, no evidence for that. That's their philosophy. Yeah,
very important.
Great job. I think we're on. How long left is this video? Praise. Oh yeah, I can see it. So it's still very long. So I think we better move on if we really want to destroy everything.
and we may just have to do parts 1 and 2 because you're right. When you get us in here being so it sucks that they had to use chromosome 2 fusion as their first, because that always requires a lot of time. So maybe
that's not their best evidence.
No,
that's not their best evidence.
No, I agree. It makes no sense that because
Actually, their best evidence are distant starlight and nested hierarchies. Their second evidence are this chromosome fusion and dating methods. So best evidence, the starlight and nested hierarchies. Second best evidences are this chromosome 2, and the dating method. So that's all.
Actually,
Let's
that's a
move
good.
on.
We're gonna do probably a dating method one in the next few days. And jungle jargon, you're good at that too, brother. So we'd love you to be there. We've done distant starlight recently. We've done nested hierarchies the other day. We're doing this one, and yeah, hopefully we'll do dating methods soon. So praise if you're still there, brother. You can
Let's
click
move on.
play at any time. You there? Praise, you there?
No, I'm not. I just got back. My bad.
Praise,
Next.
did we lose? Praise, did we lose you at the chromosome 2 fusion?
Yo,
I mean, Praise thought that.
can you hear me?
Praise's argument.
Next
Praise's
slide.
argument. Well, I mean, shouldn't we all have 48 chromosomes? But so Praise, I hope you learned something there, because never use that argument in a debate, brother.
Yeah. There's a lot of information for it out.
So you learned something there?
Yeah.
Good.
I learned. Let me see. I'm trying. This is so much that was before. I have to go back and hear again.
At least hopefully you learned why they'll say humans have 46 and
Because they have to say that.
Dummies.
Yeah, I think we have to move on. I think we have to
Find a
move
human
on.
chromosome that looks a lot like two chimpanzee chromosomes fused together. But if no such chromosome is found, then the theory would be seriously undermined. The answer comes in the form of human chromosome number 2. What we find is that chromosome 2 is very different from all the other human chromosomes. In fact, it looks exactly like two chimpanzee chromosomes glued together.
And we already refuted all this. So yeah, just keep it playing.
Well,
we're not going to
all
rehash
chromosomes
that
have
all
very
again. So just keep
specific
it playing.
Let's strike up another conversation on this, guys.
I know, but hey, the second we heard chromosome 2 fusion, we just destroyed it. Actually, you know what? Don't fast forward. Just let it play through so the audience will see the lines of evidence they use. For example, the telomeric regions. You'll see them cover all that, and you'll see how we demolish that. So let it play through, please, and then we'll get to the next one.
Every chromosome is a special region called a centromere. This unique structure is an attachment point for something called the mitotic spindle. It allows duplicated chromosomes to be pulled apart during cell division. Centromeres are made mostly of repetitive DNA sequences, and they're very easy to recognize. Normal chromosomes have and need just one centromere structure. Chromosomes also have DNA structures called telomeres at their ends. Telomeres are important because they protect the ends of chromosomes from falling apart or from fusing with other chromosomes. Like centromeres, telomeres are easy to identify. They have recognizable structures with special repeating sequences of DNA. All normal chromosomes have a single centromere, usually near the center, and two telomeres, one at each end. But chromosome 2 is different. It has not one but two centromeres, one that is functional and one that has mutated a bit and is now non-functional. It also has not two but four telomere sequences. Strange as this seems, the story gets even more interesting. These extra telomeres and centromeres are arranged exactly how you'd expect them to be if two chromosomes were stuck together head to head. They're arranged as follows: telomere, centromere, telomere, telomere, centromere, telomere. As you can see from this diagram, this is just what a combination of two chromosomes ought to look like. Also, it turns out that chromosome 2 genes and genetic material found on two chimpanzee chromosomes, 2A and 2B. So, the unique structure of human chromosome 2 completely fulfills what we would expect if humans and chimps do in fact share a common ancestor. Its odd structure is best explained by the theory of common descent. In other words, the most reasonable way to explain why chromosome 2 looks so strange is that a human-like ancestor long ago developed a mutation that fused two primate chromosomes together. Sometimes it's argued that chromosome 2 has these extra sequences because it was specifically designed that way to carry out some function that we've not yet discovered. But the vast majority of scientists agree that this is an ad hoc argument. According to our current knowledge of biology, there is absolutely no reason that a chromosome should have extra centromeres and telomeres. The most reasonable explanation is that chimps and humans share a common ancestor. Another way to test the theory of common descent is to look at special pieces of DNA called pseudogenes, or false
Praise.
genes. Basically,
Praise, pause that real quick.
Pseudogenes are
So I hope everybody's seen that she pretty much, well, we wouldn't expect there to be function there. Well, guess what? Function's been found. She said we shouldn't expect any extra centromeres, telomere-like sequences. Well, we just spent almost 45 minutes destroying the fact that they're rebuking their claim that those areas are found, and the very, very small regions that are found are all found throughout the genome, not associated with any type of fusion event. So do you see how Biologos, supposed one of their best evidence, each point that they made, we systematically demolished. And they didn't even touch on anything regarding the function. They just went right over it saying, well, we wouldn't expect there to be any function, or, it's just post hoc, ad hoc. I mean, do you realize how much function and activity really is there? So yeah, I hope everybody noticed that real quick before we move on.
Yes, I noticed.
Okay, keep going there.